Hi, my name is Michelle Trautwein, and I'm a curator at the California Academy of Sciences, and I study flies and face mites, amongst other things. So thanks for tuning in to learn more about these beasts on your body. So human bodies are this rich conglomeration of species, bacteria, viruses, fungi. And over the past couple of decades, I think we've become more used to the idea that we have diverse microbiomes in our gut and on our skin. But I think that the reality that a couple of animal species are actually a part of that microbiome as well is really still a pretty unappreciated fact. So I've been studying face mites for a few years, and even as an entomologist, it's hard to get used to the idea that we have microscopic arachnids living on our bodies. Here's one pictured here, belly up with its long tail and 8 legs and tiny mouth. Now, these mites are called face mites, but they don't just live on our face. They're actually follicle mites that live in hair follicles all over our body. So they could just as easily be called ear mites, genital mites, nipple mites, for example. And maybe you'd expect that because these mites are living on our face, that we would already know quite a bit about them. But in fact, the opposite is true. We know very little about these animals that are actually sharing our face with us. um, Here's what we do know about these mites. They're all from the genus Demodex, and Demodex are all microscopic mites that live in the skin of mammals. And so we aren't alone in having these mites. They're part of our mammalian heritage. There are about 150 described species of Demodex, and though they've been found on almost every mammal that they've been examined for. So there are likely thousands more that are yet to be discovered and described. And mammals seem to host multiple species of Demodex that live in separate microhabitats on the body. And that happens to be the case for humans. So we have 2 species of Demodex on us, aren't we lucky? The first, Demodex folliculorum here, was discovered in the mid 1800s. And we were thought to only have one species until about 50 years ago. A second species, Demodex brevis, was discovered. So folliculorum is long and skinny and lives more superficially in our pores. And brevis is this short and chunky guy over here and is thought to live within our sebaceous glands. Folliculorum often lives in clusters, so it's easier to sample. Brevis is usually found alone. So a comparative analysis of the mitochondrial genomes of these 2 mites showed that they are not very close relatives and are likely tens of millions of years divergent from each other, even though they look so similar and live in such a similar habitat on human faces. So now I'm going to tell you more about what we've learned about face mites by looking at their DNA. And I know this is a genomics seminar, and we have been working on getting a face mite genome for years, and we've just hit obstacle after obstacle. We've recently had some really great success, which I'll tell you about in a minute. But first, I'm going to tell you more about what we've learned by looking just at the mitochondrial DNA of these face mites, doing PCR and basic Sanger sequencing. So the first big discovery we made about face mites is that you almost certainly have them. In a small study that we did based on the presence absence of mite DNA, 100% of adults that we sampled had evidence of mites on their faces. And so this leads us to believe that none of us are immune. We all have face mites. And so this 100% recovery is in contrast to the recovery rate we find when we looked for whole mites, bodies of mites, which we found on only 14% of adults. And so what this means is that just because you don't see them, it doesn't mean they're not there. They can be really tricky to find. The next discovery we made was about transmission. And what we found is that your face mites are really your own. You don't share them with strangers. So face mites aren't getting casually transmitted when you socially kiss cheeks or when you sleep on a hotel pillow, but they are getting transmitted between family members, particularly between parents and offspring. So this is a likelihood tree of about 1,000 base pairs of mitochondrial DNA from folliculorum, and highlighted here are family groups, so you can see how they cluster together. Across the tree, haplotype diversity is very high, so most people sampled host mites with unique haplotypes, and when haplotypes are shared, it's amongst family members. And so this is very unlike the data, for example, from lice, where haplotypes are shared by unrelated individuals much more often. And you can imagine it's because people, head lice are kind of hopping from one head to another much more frequently, whereas face mites are really sticking to their hosts. And so we expect that you get colonized with mites from your parents or caregivers early on, and then keep them for the rest of your life. So this means that face mites are transmitted vertically instead of horizontally, like lice, which means that they are a proxy for your DNA because they're transmitted vertically, just like your DNA is from parent to offspring. And so we hope that if we sampled face mite DNA from people from all over the world, we would see structured genetic diversity and be able to retrace ancient histories of human movement. So this is a map of ancient human migration generated by human mitochondrial data from National Geographic. And this is the kind of thing we hoped we could recreate with face mite DNA. Looking to human parasites and commensals to understand our own evolution is not new. As humans evolved and moved around the world, we've harbored and transported a whole suite of species with us. For example, some of my favorite studies are focused on lice DNA. This is a beautiful head louse shown here. And these studies have allowed us to date, for example, when in our evolutionary history humans first lost their body hair, when humans started wearing clothes, and they also provided independent confirmation that we likely intermingled with other Homo species, and this is before the Neanderthal genome. These studies also showed us that we probably got our pubic lice from gorillas, which is weird. But I think the take-home message is that arthropod parasites like lice and mites, face mites, aren't just pests on our bodies. They're also really important storytellers and can reveal aspects about our own past. So we sampled people of diverse ancestries for both brevis and folliculorum, and here are two unrooted likelihood trees based on about 1,000 base pairs of mitochondrial DNA. The top is brevis, below is folliculorum. And the first thing I want you to notice is that these trees for these two very distantly related mites have the same shape. That means that they've undergone parallel evolutionary processes, which makes sense because they are living and evolving on the same host. But because brevis and folliculorum are distantly related, we considered that maybe they had had different histories of transmission to humans. So maybe one of them, humans acquired one of these species much more recently than the other, for example. But these parallel phylogenies make it seem likely that they both have been evolving together under similar conditions for a really long time. Now let's take a closer look at folliculorum. Here we find these really deeply divergent lineages structured according to host ancestry. And what that means is that different people from different parts of the world host different lineages of face mites. So we find short kind of bushes here that include virtually all hosts of European and a clear clustering of East Asian ancestry. And then we have these really long branches that include hosts of African, South and Southeast Asian, and Latin American ancestry. And what's amazing is that these lineages are persistent. So for example, we did most of our sampling in the US, where people are have a diverse, mix of ancestries. Yet, if people have lived away from their geographic region of origin for generations, they often still host face mites that are very specific to their ancestry. So this is kind of like 23andMe, but with face mites. Another really interesting thing about these results to me is that the genetic signatures here reflect aspects of historical movement by humans at two different time scales, both very ancient and very recent. So, for example, we find the highest genetic diversity on hosts of African ancestry. And that's because humans and human face mites both originated in Africa. But also, the genetic diversity of face mites in Latin America looks a lot like Africa in terms of the level of diversity. And you can see that in these pie charts here, which show the frequency of lineage recovery according to host ancestry. And so this diversity in Latin America doesn't reflect anything ancient about Latin America necessarily, but instead it reflects the more recent admixture of human populations in Latin America. And so in some important ways, these results really do mirror human evolution. But there's one notable contrast, and that's this close relationship between mite lineages that include the majority of East Asian and European hosts. So genomic estimates of the relationships between human populations suggest that Europeans and South Asians actually share more affinity than Europeans and East Asians. But we find the opposite, and so that's a little bit of an unexpected grouping. But we find that our results do closely resemble this trichotomy that's recovered between major human mitochondrial haplogroups. So there are three major mitochondrial haplogroups in humans, L, M, and N. And though they each have broad geographic ranges all over the globe, they also have geographic centers. And so we find mirror lineages to these mitochondrial haplogroups. We find African mite lineages here in yellow that are similar to human mitochondrial haplogroups L. We find South Asian and oceanic mite lineages similar to human mitochondrial haplogroup M. And we find a Eurasian lineage similar to human mitochondrial haplogroup N. Also, these groups can be defined by what they lack, and Eurasians largely lack haplogroups M and L, just as Eurasians lack mite lineages endemic to Africans and South Asian and oceanic groups. So it actually makes sense that Folliculorum phylogeny maps more closely to the matrilineal human mitochondrial phylogeny than an autosomal-based phylogeny representing both maternal and paternal genetic contributions if you think about the means of mite transmission. So as I mentioned before, Folliculorum and brevis are found in follicles on nipples. And so initial colonization of infants may take place during breastfeeding and thus have established this primarily matrilineal line of mite transmission. So now that we can see that these mite lineages reflect human population divergences, we wanted to date them to see if we could also correlate them with their timing. So dating divergences in a microscopic arthropod parasite can be pretty difficult for a few reasons. First, there's no fossil record that we can use to constrain the root of the tree. We don't have a good outgroup because we don't know the closest relatives to these face mites. And we don't know a realistic mutation rate to rely on. So previously for Folliculorum, we have done a dating analysis based on a standard arthropod mutation rate for mitochondrial DNA. And the dates between our splits ended up being unrealistically old, millions of years, which doesn't make sense for modern humans. But it's known that mutation rates for arthropod parasites can really vary. So lice workers had a similar problem, and they resolved this issue in a very elegant way. They sampled primate lice and found chimpanzee lice to be most closely related to human lice. And then to date their tree, they constrained the split between chimp lice and human lice to 5 million years ago, which is the known age of the split between the hosts. So a major goal for us is to do the same, and we expect that we inherited one, if not both of our mites, from our primate ancestors. So as ape and then hominin species evolved, our mite species just changed with us. So if we could sample chimpanzee mites, they would likely be the closest relatives to human mites, which would allow us to constrain our dating analysis. But as of now, no ape Demodex has ever been sampled or described, and we are very challenged to find a wild chimp to swab. So in the meantime, we had to try a different experiment that was inspired by work done in tuberculosis that builds on what we know about human mitochondrial lineages. So we ran 2 dating scenarios to see if under either scenario, the divergence times of mite lineages would correspond to those known for humans. So first we constrained the root of the folliculorum tree to 200,000 years ago to coincide with the origin of our species. But under that scenario, we recovered older than expected dates for the splits between African and non-African mite lineages. Next, we constrained the root of the folliculorum tree to 70,000 years ago, which is the approximate age of the origin of the Africa-centered human mitochondrial haplogroup L3 that's ancestral to all non-African populations. And rooted at 70,000 years ago, the dating estimate for mite lineages are much more in line with our current understanding of human population divergences and migrations. So this is the same tree that I showed you before, just with its branches bent around the map. And here we have origins of out-of-Africa populations at about 65,000 years ago, the divergence of Europeans at 32,000 years ago, and the most interesting one to me is the 20,000 years ago for the origin of a mite lineage that serves as a proxy for migration into the Americas. A few years ago, 20,000 years ago, would have seemed too old for migration into the Americas, but new human genetics and archaeological studies keep pushing that date back, and now 20,000 years ago seems reasonable. So now moving on to current work, my team and I have been sampling people from all over the world. This mostly happens here in the US on the public floor of the Cal Academy. This is pre-pandemic, of course. But lucky for us, San Francisco brings in visitors from all over the world. And right now we have about 2,000 samples representing people from 95 different countries, which I think is just amazing. But also, some samples have been sent to us from people living in their region of origin, including hosts from Papua New Guinea, Guam, and including a hunter-gatherer group from Namibia. And what we found as our data set grows is that the picture is messier than we previously imagined, but there's still this geographic centering of clades that reflects the distribution of human mitochondrial haplogroups. So to understand more about this system and how folliculorum varies from brevis and how, for example, African lineages may differ from Asian lineages, may differ from European lineages, and so on, we've been trying to sequence a genome, trying. And even though we're in 2021, sequencing the genome of a microscopic multicellular eukaryote that cannot be cultured has proven to be very, very difficult. So it's been done for another microscopic proto-arthropod, tardigrade, which is a creature that's a little bit larger. But in that genome study, it took hundreds of tardigrades that were grown in culture to sequence that genome. And so just to give you a review of our previous attempts at genome sequencing and a glimpse of our current success, here's a quick summary. So in terms of sampling, we've tried sequencing genomes from single mites and pooled mites. At one point, we pooled up to 300 mites to try to get a genome sequenced, and I can't overstate how hard it is to collect 300 face mites. Just the collecting, the handling time, it took a solid week. Dr. Athena Lamb at the California Academy has led the molecular work for this project, and she uses a QIAGEN microkit for extractions generally. And because we recover so little DNA, we've experimented with crushing mites, putting them through freeze-thaw cycles, leaving them for an extended time in proteinase K to get the DNA to be released from the chitin. We've tried a whole genome amplification method in building the library. We have made genome sequencing attempts on MiSeq, HiSeq, NovaSeq, and PacBio platforms, all of which failed. And what we found is that we can very easily amplify human host and bacterial DNA, but we don't get a lot of mite DNA. And when we do, it's primarily mitochondrial. So we were only slightly hopeful when we teamed up with Kristen Mars and her team at PacBio to try out a new ultra-low protocol for hi-fi sequencing. And against the odds, we've had some really great success. So this ultra-low method lets us use a starting sample of only about 5 nanograms of DNA. And for the first time ever, it looks like we've recovered enough mite DNA to assemble a genome. Folliculorum appears to have a small genome at about 50 megabases. For comparison, Drosophila melanogaster is 175 megabases. Humans are 3 gigabases. And this makes sense for a parasitic arthropod that is it's an obligate parasite that lives in the body of another organism. It makes sense that their genome is quite reduced. In previous attempts, we've recovered very little mite DNA, like 5,000 sequences out of millions. But here, about 40% in this new PacBio genome, about 40% of our reads are face mite, 30% are human, and 30% are bacteria. Our genome assembly was done by first filtering out human and bacterial reads to avoid making chimeric contigs. And another sign of success comes from an analysis of BUSCO genes, or single copy orthologs, across arachnids. So a set of about 3,000 arachnid orthologs. From that set, we recovered about 90% of those genes, which to me is just amazing. In our previous attempts, we recovered no arachnid BUSCO genes. So we're really excited about this new genome and the possibilities that it opens up for us. So moving forward, we have preliminary analyses already done about the face mite microbiome. We're interested in developing a probe kit for diagnostics so we can more easily tell what lineages a particular host carries. And we're really interested in characterizing the genomic changes between mite lineages hosted by various human populations. So here are some of the many people that have helped me with this work. Particularly, I want to thank Dr. Athena Lamb, Matt Van Dam, Jim Henderson, and also Kristen Mars and her team from PacBio for doing an amazing job with this near impossible genome.